Good morning. Again, I'm here with you to discuss about the role of intestine. So, what is the objective of discussing the role of intestines? Because if you're discussing about the role of intestine, you can understand about the different types of the enzyme and secretion is secreted through the enzymes that will through the intestines that will help in the digestions. We already discussed about the morphological structure, that means anatomy and physiology of the intestine. So how the metabolism, how the digestions will occur in the intestines, we can see. Okay, it will make easy to understand about the intestine function. It will make easy to understand about the different secretions secreted by the intestines and the digestive activity occur in the intestines. Now come to the point, the role of intestinal juice. So in the intestines, there is a juice secreted. There is a secretion is secreted that will be called the intestinal juice. Like the secretion secreted in the gastric, that will be called the gastric juice. Similarly, secretion secreted in the intestine will be called the intestinal juice. So about 1 to 2 liter of intestinal juice, a clear yellow fluid, are secreted each day. So per day, approximate 2 liter up to 2 liter juice will be secreted through the intestines. That juice is the, it can be enzymes, it can be some other things. So it will be the intestinal juice. So the intestinal juice will contain the water and mucus and is slightly alkaline. That is, the pH is 7.6. So this juice, this intestinal secretion will contain the water and mucus. Together, pancreatic and intestinal juice provides a liquid medium that adds the absorption of substance from the chyme in the small intestine. Pancreas is also secreted the pancreatic enzymes into the duodenal, into the intestine. So with the intestinal juice, the pancreatic juice will mix and provide a liquid medium that will help in the absorption of the substance. The absorptive cell of the small intestine synthesize the several digestive enzymes called the breast border enzyme and insert them into the plasma membrane of the microvilli. So some enzymatic digestion occur at the surface of absorptive cells that line the villi rather than in the lumen exclusively as occur in the other part of the gastrointestinal tract. Among the breast border enzymes are four carbohydrate digestive enzymes. These breast border enzymes contains the different types of the enzymes, in which the four carbohydrate digesting enzymes called dextrinase, maltase, sucrase, and lactase. So these are the four different types of enzyme that will help in the digestion of the carbohydrate: dextrinase, maltase, sucrase, and lactase. Protein digesting enzymes here that will be called peptidase. It aminopeptidase and dipeptidase. So this peptidase will helping into digestion of the proteins. And two types of the nucleotide digesting enzyme, that will be the nucleosidase and phosphatase. So it will be help to digesting the nucleotides. Also, absorptive cell slough off into the lumen of the small intestines. They break part and release the enzyme that help to digest the nutrients in the chyme. Now, how the mechanical digestion occur in the small intestine? Two type of the movements of the small intestine. It's a mechanical digestion, not a chemical digestion. So try to understand which type of the two mechanical digestions. So here is the movement is there, small intestines. Segmentation and the types of the peristalsis called the migratings modality. Complexes are governed mainly by the mesenteric plexus. Segmentation are localized mixing contraction that occur in portion of the intestine distinct by the large volume of chyme. Segmentation makes the chyme with the digestive juices and bring the particles of food into the contact with the mucosa for absorption. So in the intestine, there is a different segmentation localized in the intestinal cells, and mixing contraction occur in the portion of the intestines distinct by the large volume of the chyme. So this segmentation mix the chyme with the digestive juice and bring the particles of food into the contact with the mucosa for absorption. They do not push the intestinal content along the tract. A segmentation starts with the contraction of circular muscle fiber in a portion of the small intestine, an action that constricts the intestines into the segments. Next, muscle fibers that encircle the middle of each segments also contract, dividing in the each segments again. Finally, the fibers that first contract relax, and each small segment unites with and adjoining the small segments, so the large segments are formed again. So these small segments is going to form and make other large segments. As this sequence of the events repeat, the chyme sloshes back and forth. Okay, so during this process, the chyme will go on to backward and forward movement. So the segmentation occur most rapidly in the duodenum, about 12 times per minute, and progressively slow about the 8 times per minute in the ileum. This movement is similar to the alternatively squeezing the middle and then leads and the ends of the capped tubes of toothpaste. Now understand, this movement is similar to alternately the squeezing the middle and then ends of a capped tube of toothpaste. Now you can understand, when you put the toothpaste, pressure on the toothpaste in the middle, you can say there is the toothpaste will come out, and when you release the pressure, it will be goes back. So this is the coming backward and forward movement help in the in the mixing again. After most of the meal has been absorbed, which lessen the distension of the wall of the small intestine, segmentation stops and the peristalsis begin. So most of the meal has been absorbed after that. Okay, after most of the meal has been absorbed, so when the meal is going to absorb, that means the distension of the wall is going to reduce. So the lessen distension wall of the small intestine, and segmentation will stop, and then peristalsis will begin. The type of peristalsis that occur in the small intestines termed as the migrating motility complexes. Okay. So this peristalsis in the intestine will be called the MMC, migrating motility complex. Begin in the lower portion of the stomach and pushes the chyme forward along a short stretch of small intestines before dying out. The MMC, this motility migrating complex, slow the migrate down the small intestines, reaching the end of the ileum in 9 to 120 minutes. Then another MMC begin in the stomach. All together kinds remain in the small intestine for 3 to 5 hours. Now these are the cells and the small intestine mucosa. They are the cell types, location in the mucosa, and their function. For example, the absorptive cell, it will be located in the epithelium and intestinal glands. Function is the digestion and absorption of the nutrients in chyme. Second cell type is the goblet cells. That will be the epithelium oblique intestinal glands. Function is the secretion of mucus. Paneth cell is there. Paneth cell we already discussed about. It will be helping to remove the bacteria. It will present into the intestinal glands, and secretion of the bactericidal enzyme, lysozymes, and phagocytosis. In the process of phagocytosis, it will be work. It will be help. G cell, that is the gastrin cell or G cell, intestinal glands of the duodenum. Now, what is the function of the G cell? Secretion of the hormones intestinal gastrin. I cell is there. It will again present in the duodenum. And what is the function of the I cell? Secretion of the hormone cholecystokinin, which stimulate the release of pancreatic juice and bile. So I cell will secrete the hormone the hormone cholecystokinin. So when the cholecystokinin will be secreted, it will causes to enhance, to increase the release of pancreatic juice and bile. It will causes the stimulate the pancreatic juice and bile. So most important, we have seen the Paneth cell that will act as a phagocytosis. Goblet cell will be secrete the mucus. G cell secrete the gastrin hormone. I cell secrete the cholecystokinin, and cholecystokinin stimulate the pancreatic juice and biles. K cell is there. It will again present in the intestinal glands, secrete the hormone glucose dependent insulinotropic peptide, which release the insulin. Okay, so K cell is here. If the K cell, that will causes the ultimately the release of the insulin from the pancreas. M cell is there. It will present in the intestinal glands of the duodenum and the jejunum. And the secretion of the hormones insulin, hormone motilin is there, which accelerate the gastric emptying. So by M cell, by secretion of the hormone motilin, which enhance the gastric emptying, stimulate the intestinal peristalsis, and stimulate the production of pepsin. S cell is there. S cell is act like the you can say secretion of the hormone secretin. So S cell is enhance the secretion of secretin and present in the intestinal glands. Again, these are the name of the enzymes, their source, their substrate, and the products. Like saliva is there. In the saliva, salivary amylase and lingual lipase is there. Salivary amylase uh, secreted by the salivary glands, and their substrates is about the starch, and the product is about the maltose. Similarly, lingual lipase is their source is the lingual gland in the tongue. So what is the whatever is the lingual gland present in the tongue, that will be secreted the lingual lipase, and the substrate is the triglyceride, and product is the fatty acid and diglyceride. Similarly, second will be the gastric juice. Pepsin is there, activated form of pepsinogen by pepsin and hydrochloric acids. Source is the stomach chief cell. Protein is the substrate and product is the peptides. Gastric lipase is there. It will be also released by the chief cells, and their substrate is the triglyceride, and product is the fatty acid and the monoglycerides. Pancreatic amylase is there, that will be released by the pancreatic acinar cell, secreted by the pancreatic acinar cell. Similarly, these are their substrate and the products. Trypsin is there, activated form of the trypsinogen by enterokinase. So when the trypsinogen will be released, you will see in the later slides, trypsinogen will be released, and trypsinogen from the intestine, when trypsinogen will be released, it will interact with the enterokinase present in the intestine. So it will causes the activation of the trypsinogen, and it will become the active trypsin. Release where pancreatic acinar cell. So pancreas will release the trypsinogen in active form, and it becomes the active when reaches to the intestine in the form of trypsin. Chymotrypsin is there, pancreatic acinar cell, activated form of the chymotrypsinogen by trypsin. Elastase is there, it will be also released by the pancreatic acinar cells, activated form of the proelastase by trypsin. Carbopeptidase is there, activated form of the procarboxypeptidase by trypsin. Pancreatic lipase is there, it will be also released by pancreatic acinar cell. So all these things are released by the pancreatic acinar cell, including the nuclease is there, ribonuclease and deoxyribonuclease. Okay, so pancreatic juice, especially pancreatic acinar cell, will secreted the pancreatic amylase, trypsin, chymotrypsin, elastase, and pancreatic lipase and nuclease. Breast border present in the intestine. So small intestine having the breast border, and it will be like the alpha dextrinase. It will be released by the small intestine. Maltase, sucrose, lactase, enterokinase, peptidase, this means aminopeptidase and dipeptidase, nucleosidase and phosphatase. These all be secreted by the small intestines. Now, chemical digestion in the small intestine. After the mechanical digestion, come to the point new is the chemical digestion in the small intestine. So, in the mouth, salivary amylase convert the starch to a maltose, means polysaccharide to a disaccharides. Starch is a polysaccharide converting to the maltose to disaccharides, maltriose a trisaccharide, a dextrins a short chain branched fragments of starch with 5 to 10 glucose units. Sorry. In the stomach, pepsin converts the protein to the peptides, the small fragments of the proteins. And the lingual and gastric lipase convert the some triglyceride into the fatty acid and diglyceride and monoglycerides. Thus, chyme entering the small intestines contains the partially digested carbohydrate, proteins, and lipids. The completion of the digestion of carbohydrates, proteins, and lipids is a collective effort of pancreatic juice, biles, and intestinal juice in the small intestine. So the completion of the digestion of carbohydrates, so it will be gonna went to the end of the digestion. The completion of the digestion of carbohydrates, proteins, lipids. These all the collective effect of the all juices. Pancreatic juice is there, bile juice is there, intestinal juice is there. So when we are talking about the end of the digestion, so end of the digestion is just by the combined effort of the every secretion happenings during in the bodies. All the chemicals and mechanical phases of digestion from the mouth through the small intestines are directed toward changing the food into the forms that can pass through the absorptive epithelial cells lining the mucosa and the underlying blood and lymphatic vessels. These form of these form are the monosaccharides like the glucose, fructose, galactose from carbohydrates, single amino acids, dipeptide and tripeptide from proteins, and fatty acid, glycerol and monoglycerides from triglycerides. Passes of these digested nutrients from the GIT tract into the blood or lymph is called the absorptions. We know the absorptions. So these digested nutrients can goes into the blood through the GIT. So absorption. So absorption of material occurs via diffusion, facilitated diffusion, osmosis, and active transport. Now we know the diffusion, facilitated diffusion. Diffusion is just process of going through the plasma membrane. These nutrients will goes into the blood, simple diffusion process. Facilitated diffusion, there is some carrier will be there, so that will facilitate the diffusion. Some factor is there, so that will enhance to the diffusion, facilitated diffusion. Then osmosis and active transport is there. About 90% of all absorption of the nutrients occur in the small intestine. The other 10% occur in the stomach and large intestine. So small intestines playing the absorption of the nutrients. How much? 90%. And the remaining 10% occur in the some small amount of the absorption will occur in the stomach as well as in the large intestines. So any undigested or unabsorbed material left in the small intestines passes on the large intestines. You can see here there's the absorption of the digested nutrient in the small intestine. So for simplicity, all digested foods are shown in the lumens of the small intestine. So you can see there's a lumen of the small intestine. So these all are the showing the digested foods. Okay. Even though some nutrients are digested by the blood enzyme also. You can see there's a glucose and galactose. So secondary active transport with the sodium, they are gonna to secretes. Fructose is there by the facilitated diffusion will be gonna into the facilitated diffusion, it will goes into the blood capillary, and in between you can see there's the epithelial cell of the villus. There's a microvilli is present on the epithelial cell surface. Amino acid there by the active transport or secondary active transport is gonna to amino acid inside and causes the diffusion and will be goes into the blood. Dipeptide and tripeptide goes to the secondary uh, transport mechanism. Short and fatty acid will go through the simple diffusion process. It will causes this, go through the simple diffusion into the your epithelial cell and then passes to epithelial cells basolateral membrane, basolateral surface, and goes into the blood. Large intestine is the terminal portion of the gastrointestinal tract. We know from the starting from the mouth going to the esophagus, stomach, and small intestine and large intestine. So large intestine is the end part, is the terminal part of your GI tract. The overall function of the large intestines are completion of the absorption, the production of certain vitamins, the formation of the feces, and the expulsion of the feces from the body. So it will help in the complete the absorption, because 90% absorption is completed from the small intestines. Other amount of this absorption is completed into the stomach. So remaining completed by the terminal portion, that is the large intestine. So absorption will be completed, production of the certain vitamins will be here, formation of the feces will be here. So when the water will be reabsorbed through the large intestines, the waste material will be solid, that will be the feces, and the expulsion of the feces from the body. Now, the large intestine anatomy we're talking about, the large intestine, which is about 1.5 meter long and 6.5 centimeter in the diameter, extend from ileum to the anus. It is attached to the posterior abdominal wall by its mesocolon, which is a double layer peritoneum. Structurally, the four major regions of the large intestines are the cecum, colon, rectum, and anal canal. Basically, you will see this in this the cecum, colon, and rectum, and the last one is the first, the anal canal. The opening from the ileum into the large intestine is guarded by the fold of mucous membrane called the ileocecal sphincter. Now, the small intestine, the ileum part, is going to connect with the large intestine, the first part. Okay, so mucous membrane, there's a sphincter that will be called the ileocecal sphincter, because it's going to connect with the cecum. So ileum and cecum having a sphincter, a wall, that will be known as the ileocecal sphincter, which allow the material from the small intestine to pass into the large intestines. Hanging inferior to the ileocecal wall is the vacuum. Sorry, is this wall is the cecum. So hanging inferior to the ileocecal wall with a cecum, a small pouch about the 6 centimeter long. Attached to the cecum is a twisted coil tube measuring about 8 centimeter in length called the appendix. Yeah, vermiform appendix, you know about the name of the appendix. The mesentery of the appendix called the mesoappendix attaches to the appendix to the inferior part of the mesentery of the ileum. The open end of the cecum merges with a long tube called the colon. The food passes the colon, which is divided into the ascending, transverse, descending, and the sigmoid portion. Both the ascending and descending column are the retroperitoneal. If you see the structure of these column in this ascending and descending column, you will be see likely goes to the back. So it will go to the retroperitoneal, and the transverse and sigmoid column are not. True to its name, the ascending column ascends on the right side. If you're talking about true to its name, so we can say the ascending column ascends on the right side of the abdomen, reaches the inferior surface of the liver, and turns the abruptly to the left to form the right colic flexus. The colon continues across the abdomen to the left side as the transverse colon. It curves beneath the inferior end of the spleen on the left side as the left colic, and passes inferiorly to the level of the iliac crest and the descending colon. The sigmoid colon, that the associate colon is, begins near the left iliac crest, project medially to the midline, and terminate at the rectum at about the level of the third sacral vertebra. The rectum, the last 20 centimeter of the gastrointestinal tract, lies anterior to the sacrum and coccyx. These are all the just like the position and the anatomy. The terminal is about 2 to 3 centimeter of the rectum is called the anal canal. So the mucous membrane of the anal canal is arranged into the longitudinal folds. Now you can see the structure here, the large intestine. There is a cecum. Below the cecum, you can see the vermiform appendix is there. Then ileum and cecum gonna to meet at the point that will be having the ileocecal sphincter valve is there. Then ascending colon is there, transverse colon is there, descending colon is there, and sigmoid colon is there. And last is the anal canal, rectum. So If you see the structure, function of the large intestine, you can understand these are the hostile churning, peristalsis, and mass peristalsis drive the content of the colon into the rectum. Bacteria in the large intestine convert the protein to amino acids, break down the amino acids, and produce some B vitamins and vitamin C, absorbing some water, ions, and vitamins, forming the feces, and then defecating. That means empty the rectum, move out the feces. So these are the function of the large intestine. If you go through the histology, you can see there are the several notable differences between the wall of the large and small intestine. So in the small intestine, we have seen the four layers. Now in the large intestine, there would be a little bit difference between the small and large intestine wall. A few enzyme secreting cells are found in the wall of the large intestines, and there are no circular folds or villi. In the large intestines, no circular folds, no villi. Other than in the anal canal, the mucosa of the colon is simpler columnar epithelium made mostly of the enterocytes, absorptive cells, and goblet cells. In addition, the wall of the large intestine has far more intestinal glands which contain a vast population enterocytes and the goblet cell. These goblet cells secrete the mucus that ease the movement of the feces and protect the intestine from the effect of acids and gases produced by the entering bacteria. Now understands. There is a goblet cell present in the intestine, large intestine. These goblet cells will secrete the mucus. So mucus will gonna to lubricate all the passes of the intestines. So it will easiest to the movement of the feces. So the feces can be easily move in the large intestines, and also it will protect the intestines wall because there is a bacteria that will causes the infection, that will causes the produce some acidic environment is there. So in that cases, these goblet cell producing mucus will help them. The enterocytes absorb the water and salt as well as the vitamins produced by your intestinal bacteria. So these enterocytes will helping into the water present in the large intestines, salt present in the large intestine, as well as the vitamins or nutrients. Whatever is present in the large intestines can be absorbed by the enterocytes. Now, which type of the mechanical digestion is going to happen inside this? Mechanical digestion. After the chemical digestion, we will see on the mechanical digestion. The passage of chyme from the ileum into the cecum is regulated by the action of the ileocecal sphincter. We know the ileum and the cecum are connected at a point having a wall that will be known as the ileocecal sphincter. And this sphincter will allow the pass of the food from the small intestine to large intestine. That means from the ileum to the cecum. Normally, the valves remain partially closed, so the passage of chyme into the cecum usually occurs slowly. Partially closed, not fully closed. These valves are partially closed, so a small amount of the chyme can be passed from the intestine to the large intestine, from the small intestine to the large intestine. So slowly manner, it will passes a small amount of the food and very slow from the ileum to the cecum. Immediately after a meal, a gastroileal reflex intensifies the peristalsis in the ileum and forces the any chyme into the cecum. So when the peristalsis will be enhanced, these forces will be enhanced, the large amount of the food can be passed. Hormone gastrin also relax the sphincter. If release of gastrin will causes the relax of the sphincter, that means more food can be passed from the small intestine to the large intestine. Whenever the cecum is distant, the degree of contraction of the ileocecal sphincter intensifies there. So the movement of the colon begin when substance passes the ileocecal sphincter. So when the substance will pass from small intestine to large intestine, or you can say when the when the foods will reaches into the large intestines, then colon movement will be start. Now, because chyme moves through the small intestine at a fairly constant rate, The time required for a meal to pass into the colon is determined by the gastric emptying time. Now you can understand, when the gastric emptying, that means when the stomach will pass the food, food goes into the intestine, then intestine will pass the food, food will goes into the large intestines. So if the stomach empty time is delayed, the stomach will pass the food into the small intestine, but in a delay manner. So obviously food will not reaches into the large intestine. So these all things will be depends upon the gastric emptying, how much time taken by the stomach to empty themselves. Okay. As food passes through the ileocecal sphincter, it fills the cecum and accumulate in the ascending colon. So after the cecum, it will goes into the ascending colon area. So one moment characteristic of the large intestine is the hostile churning. Now, what is the hostile churning? You know, this ascending colon, that means food has goes against the gravity. So hostile churning is there. In this process, the hostel remain relaxed. The hostel is the area of the large intestines. Okay, so this hostel will remain relaxed and become distinct while they fill up. Okay, so it will be relaxed and it will be distinct. So when the food will come into the large intestine, it will fill in the hostel. So when the distension reaches at the certain points, the wall contract and it squeeze the content into the next hostel. So when the hostel will fill with the fluids, now it will be contract and this food will be passed into the next hostrum. So similarly, next hostrum will be going to fill with the foods and the next hostrum will be again contract. This food will pass into the for the next hostrum. So this process, the food can goes to forward. So peristalsis also occur, although at a slower rate than in more proximal portion of the tract. A final type, a final type of the movement in mass peristalsis, a strong peristaltic wave that begins at about the middle of the transverse colon and quickly drives the content of the colon into the rectum. So there is after these all incidents, the food goes into the ascending colon, the transverse colons, then goes to the descending colons. A final type of movement is a mass peristalsis, a strong peristaltic wave that begin about the middle of the transverse colon. So it will start at the middle of the transverse colon and quickly drive the content of the colon into the rectum. Because food in the stomach initiate the gastrocolic reflex in the colon, mass peristalsis usually take place 3 or 4 times a day during or immediately after a meal. So during the meal or after the meal, this mass peristalsis usually take place 3 to 4 times a day. Uh, there is a certain quiz I put here that will help to all of you. Okay. Uh, Before quiz, I will explain one more thing again, the chemical digestion in this. And then after chemical digestion, we will be page. The final stage of digestion occur in the colon through the activity of the bacteria that inhibit the lumen. So mucus is secreted by the glands of the large intestines, but no enzyme are secreted. Chyme is prepared for the elimination by the action of bacteria, which ferment any remaining carbohydrates and release of hydrogen, carbon dioxide, and methane gases. These gases contribute to the flatus gas in the colon and term the flatulence when it is excessive. That is the reason we expel the gases. Okay, that is called the flatulence. Okay, so these gases will contribute to the flatus. Which type of the, because chyme is prepared for eliminating by the action of bacteria which ferment any remaining carbohydrate. So during this process, there is some gases will be developed like the hydrogen, carbon dioxide, and methane gases, and it will be cause the flatulence or flatness. Bacteria also convert any remaining proteins to the amino acid and break down the amino acid into the simpler substance like the indole, scatol, hydrogen sulfide, and the fatty acid. Some of the indole and scatol is eliminated in the feces and contribute to their order. The rest is absorbed and transported to the liver, where these compounds are converted to the less toxic compound and excreted in the urine. So the you can say the means odor, the bad odor you are smell, that will be due to the presence of the indole and escatol, okay, in eliminated in the feces. So bacteria also decompose the bilirubin to simpler pigments, including like the stercobilin, which gives the feces their brown color. So the feces color is due to the presence of stercobilin in the feces, okay, and the feces odor is due to the indole and escatol. Bacterial products that are absorbed in the colon include the several vitamins needed for the normal metabolism, like the vitamin B and vitamin C. Or vitamin K is there, especially. Okay, some form of the vitamin B and vitamin K is there. By the time chyme has remained in the large intestine 3 to 10 hours, it has become solid or semi-solid because water absorption and is now called the feces. Water is absorbed from the large intestines. Now this become the semi-solid mass, and that will be called the feces. Chemically, feces consist of water, inorganic salts, slough of the epithelial cell, from the mucosa of the GIT tract, bacteria, products of the bacterial decomposition, unabsorbed digested material, and indigestible parts of the food. So those things that is not digested, maybe it is unabsorbed digested material or indigestible food, these all can be passed through the feces. Now, these are some summary of the digestive activity. Like the structure, you can see there's a lumen, mucosa, and muscularies. In the lumen, the bacterial activity will be there. Breakdown of the undigested carbohydrate, proteins, amino acid into the product that can be expelled in the feces or absorbed and detoxified by the liver. Synthesizes the certain B vitamins and vitamin K. Mucosa secrete the mucus and lubricate the colon, protect the mucosa. Absorption is there. Water absorption solidifies the feces. So when the water will be absorbed, the feces will be solidified, semi-solid, and contribute to the body water balance. Solute absorbed includes iron and some vitamins will be there. Then the structure muscularies, in which the hostile churning will be there, that moves the content from the hostrum to the hostrum by the muscular contraction. Peristalsis is there, moves content along length of the colon by contraction of the circular and longitudinal muscle. Mass peristalsis is there, forces content into the sigmoid colon and rectum 3 to 4 times per day. And the defecation reflexes. So eliminate the feces by contraction in the sigmoid column and rectum. So the feces can be eliminated. That is called the defecation process. Now come to our quiz section. Yes. Now the first question of the quiz is the which of the following is an active enzyme? Which of the following is an active enzyme? So there is a name of the enzyme like the procarboxypeptidase, pepsin, telophase, and trypsinogen. These all the name of the enzymes, and which one is the active, because we know trypsinogen is not active. When it will meet to the press border introkinase, then it will be active in the form of the trypsin. Telophase is still not active, and procarboxypeptidase is still not active. So what is the active form? Pepsin. When the pepsinogen is will meet with the HCl, it become the pepsin. So it will be the active form. So answer B is correct. Next question. The liver contains the leaky capillaries known as the sinusoids. Okay. This enable what liver products to enter the bloodstream? Of which one enter to the bloodstream by this leaky sinusoids? Angiotensinogen is there, Kupffer cell is there, plasma protein is there, and cholesterol is there. So the right answer will be C. A plasma protein can enter into the bloodstream. Next question is the which of the following terms is used to describe the changing of large food molecules into small molecules? Very easy questions. You can understand. First, understand the questions. There are the following terms given A, B, C, and D. Now, which one is used to describe the changing of large food molecules into the small food molecules? That means by one process, like you can say, first one is the mechanical digestion. By mechanical digestion, you can convert the large food into small foods. By deglutition, by segmentation, by hydrolysis. Okay, mechanical digestion is a little bit can be okay. Large food molecules into the small molecules when the mechanical digestions. Okay, like you can say teeth is cutting the food into the small molecules. Like the motion, peristaltic movement will also break down the small foods, but it cannot be break down the every food molecules into the simpler, smaller molecules. Decrotation is also same. Segmentation is also cannot. So hydrolysis is there. So hydrolysis can convert the larger food molecules into the smaller molecules. So answer D is correct. Next question is, if blood glucose is high, what does the liver do about it? Now, blood glucose goes to higher. That means hyperglycemia chances will be there. In the blood, glucose level is higher. So how liver can reduce it? By converting the glucose to glycogen like that. Similar to something. So see, there's a four answer. The first, liver convert the glucose to glycogen or triglyceride. B, the liver performs the glycogenolysis. C, the liver performs the gluconeogenesis. D, the liver transminate glucose to produce the amino acid. Now, if we see answer number D, the liver transminate the glucose to produce amino acids, so it will be not help. C, the liver performing gluconeogenesis. No, it will again going to production more glucose. D, the liver perform the glycogenolysis. It will be help to light the glycogens, but not glucose cannot be reduced in the blood. So the liver converts the glucose to glycogen. Yeah, it is okay. The liver will convert the glucose to glycogen, and this glycogen can be stored into the liver. So when the blood glucose level is reduced in somehow coming time, the liver can again convert this glycogen to glucose and give to the blood. So again the blood glucose level is going to increase. So that is correct. So answer A is correct. Which of the following could not be used to describe pepsinogen? Which of the following could not be used to describe pepsinogen? But pepsinogen इसमें से क्या नहीं है? It is a protein, it is a hormone, it is a related to an enzyme, it is inactive. See, answer number A. It is a protein. Yes, protein. Pepsinogen is a protein. So our question was, which of the following could not be used? But protein can be used because pepsinogen is a protein, so we can define pepsinogen as a protein. Yes, okay. It is a hormone. No, pepsinogen is not a hormone. So we cannot define the pepsinogen that it is a hormone. So B is the could be answer. Now let's further we can C and D. Maybe more even accurate answer will be there. Answer number C, it is related to an enzyme. Yes, pepsinogen is related to an enzyme. So it will also incorrect. D, it is inactive. Yes, definitely. Pepsinogen is inactive. When it combined with the HCl, which will be active, pepsine. So yes, all three are incorrect. Then answer B is correct. Now the last question after this quiz. Which section of the gut perform the majority of the digestion of the food and absorb of the digested product? Very interesting and very easy. We already learned in this last slides. Which section of the gut? Now gut having the several section. Now which section we are talking about that perform the major digestion of the foods and absorption of the foods products? So the section is responsible for the digestion as well as the absorption. Which one can be possible? First, stomach and duodenum is there. Jejunum and ileum, ascending colon and transverse colon, duodenum and ileum. I know you know the answer about it. First, stomach and duodenum. Is it possible for the stomach and duodenum to make the most digested and absorbed? We know 90% is absorbed from the ileum, that is the small intestine. Another 10% is absorbed from the stomach and large intestine. So stomach and duodenum both will not work. Okay. B. Jejunum and ileum. Jejunum is a empty fold. Ileum is okay, but jejunum is not okay. So I think B is again incorrect. Ascending colon and transverse colon, these are the part of the large intestine. And large intestines will only play the 10% absorption of the nutrients. So ascending colon and transverse colon, again it is incorrect. So what is the correct? Then the D will be correct. D is the duodenum and jejunum. Maximum absorption of the small intestine occurred by the duodenum and jejunum. So D is correct. Now here are some reference book. You can follow the reference book for going to detailed study. It will help a lot. Goes through the all the digestive activity, the anatomy of intestine, function of the intestines, but that means physiology of the intestines. Now we know the intestines can be divided. If you brief, seeing the intestines can be divided into two parts: large intestine and small intestine. Small intestine having the duodenum, jejunum, and ileum. Large intestines, you can say cecum, colon, and rectum, and the last is the anal canal. So in this process, we know all the things about the intestines. What is the mechanical digestion? What is the chemical digestion? That means how mechanical digestion occur. It will help in the digestion and absorption. How chemical digestion will help in the digestion and absorption of the small intestine. Similarly, we see how mechanical digestion help, how chemical digestion will help. Okay, so these mechanical chemical process both are involved in the small intestine as well as the large intestine. So ultimately in the all alimentary canal, that means from the mouth to the anus. Mouth is the our first starting point, and the anal is the terminal point. So the food will passes from mouth to the anal canal. That will be called the all the digestive process, all the digestive system. So go through the books and see in the details about these role of the intestine in the digestion and absorption. Because if you study the digestion, if you are going to study the digestion, then you cannot leave the intestine, because intestine is the prime part where digestion occur. Except that digestion start from the mouth, it is correct. Digestion is still happening in the stomach, it is also correct. But without the function, without knowing the function of the small intestine and large intestine, you cannot complete it. So go through the books. These are the reference book. You can go through these books, read about these things, and the doubts in your minds will clear. Okay, even if you have a little bit doubt, you can ask your doubt to me, you can send your queries to me. I will solve your problem. So till that, thank you. Thank you so much for your patience listening. Then we will meet again in the next class. Till then, thank you.